Welcome and thank you for joining us again for another exciting webinar. I am Carlos Bosques, CEO at the Food Allergy Science Initiative, and I am very excited to welcome you to the seminar today, which is sponsored by Genentech. We are very thankful for the kind sponsorship by Genentech to make this webinar possible. As some of you may already know, it is our 10-year anniversary. FOSSI started in 2016 and has done amazing work pushing the boundaries of the food allergy research, thanks to our investigators. And to celebrate this great momentum, we will have a FOSSI summit, the FOSSI 10 summit, which will be celebrated on October 6 to 7 at the Broad Institute in Cambridge, Massachusetts. We hope you can join us, and we are now open. The registration is now open, so make sure that you register soon. The spaces are running out, and we hope you can join us to celebrate this important moment. At FOSSI, our focus is to understand, translate, and intervene with food allergies. And as a leader of the food allergy research, we're dedicated to advancing breakthrough discoveries in research to transform the lives of people with food allergies. And while food allergies impact millions of people across the world, understanding the disease and the availability of effective treatments remain a limitation. This year, as we prepare for an exciting series, the allergy equation clinical trials that shaping the future of food allergies, we're very excited to have put together a very great lineup of speakers that are providing information about different treatments and therapeutics that are advancing through the clinic or that are now in the market for the treatment of food allergies. Whether you're a patient, a clinician, a researcher, or just curious about food allergies, we hope these seminars provide helpful information to all of you. Before we begin, I would like to clarify that FOSSI is science-driven and evidence-based, and therefore, throughout the series, our intent is not to promote any products or companies, but instead to inform our audience on recent clinical development efforts for food allergies, and also to have the opportunity for everyone to ask questions to better understand the benefits and the challenges of different emerging options. Today, I am thrilled to be joined by a good friend, Wayne Scheffler, Dr. Wayne Scheffler, who's a FOSSI principal investigator. He's also an associate professor of pediatrics at Harvard Medical School, and he's a director of the Food Allergy Centers at MGH. Dr. Scheffler's research focus on a multidisciplinary approach for clinical programs focusing on the characterization of the immune response to food allergens and the early stages of allergen-induced immune reactions. Dr. Scheffler is doing all kinds of research focusing not only on food allergies, but also in EOE, and covers a lot of areas of research in those specific areas. Dr. Scheffler received his MD PhD from New York University and his pediatric training from Albert Einstein College of Medicine. And he also have the allergy and immunology training at Mount Sinai School of Medicine. So, Wayne, it's a pleasure to see you again. I want to remind our audience before we get into this that we'll be taking questions on the Q&A box. So please feel free to submit your questions, and we'll definitely address them at the end of the discussion. But Wayne, great to have you here.
Thanks, Carl.
Always exciting to have the opportunity to learn from you. So very excited about this seminar.
Thanks for having me, Carlos. Great to be here.
Thank you. And Wayne, I know hopefully a lot of people know Wayne for all the amazing work that he's doing, not only research, but the impact he's having directly treating patients. But I always ask the questions to our guests. How do you get here? What was the motivation to do what you do, Wayne? If you can tell our audience a little bit more about you and how you got to where you are.
Yeah. Oh, that's fun. Well, I'll try to keep it short. I was not someone who thought I would go into medicine, sort of through college. I think a lot of my colleagues who are physicians kind of see that as a calling from pretty early on. Um, you know, got really interested in biology and then immunology and actually was given the opportunity to work in a lab as an undergrad in a immunoparasitology lab and so really got excited about immunology. I won't share how exactly long ago that was, but for example, when I was in the lab, IL-10 was cloned, so it's been a while. Um, and that arm of the immune response, the sort of Th2 bias of that, as it's the jargon term, kind of the same sort of characteristics of the immune response that we also see in a lot of allergic disease, kind of got me interested. And then as I got more and more interested in clinical medicine, in part from the influence of those years and the people I met there and people encouraging me to consider getting an MD and not just pursuing a PhD. Um, I began to sort of see the epidemic of allergic disease. And I met Hugh Sampson at Mount Sinai, who recruited me, to fellowship there. And there was a mucosal immunologist, Lloyd Mayer, that was a co-mentor as well. And so that's kind of how things got started. It was sort of my interest in immunology and then sort of seeing this rise of allergic disease, including food allergies.
And we're thankful and we're actually appreciate that you ended up here because we need people like you to tackle this big problem of food allergies. Um, so Wayne, I know the title of your talk is Biting Back at Allergies, OIT, and Other Therapies. Uh, Great topic. I think we have talked a little bit about oral immunotherapy in previous webinars, but we're seeing an emergence of other therapies. Before we get into Solaire, where there's perhaps most experience and data right now. Can you comment a little bit more about the emerging therapies and that are being explored also with oral immunotherapy? You've done a lot of work in this area, so I think our audience will benefit from your information and your thoughts on this.
Sure, thanks. I did bring up a couple of slides to help illustrate, a little bit of this, and I thought, I might just, not knowing the full diversity of the audience, remind everyone of, what we mean by immunotherapy for food allergy, because it sounds fancy, but it's sort of embarrassingly 19th century. We're really just exposing people, to the allergen in a safe way and sort of hoping for an immune response that has benefit to the patient. And, the origins of immunotherapy for allergy are as old as vaccinology. And are beginning to advance in more rational directions. But as it's practiced right now, oral immunotherapy is really not what we might call a rational intervention. It's sort of exposing people to small amounts and then looking for improvements in clinical tolerability. And that has been evaluated in a few different ways. The strongest evidence we have for OIT's benefit really is in the form of showing that it supports what we tend to call desensitization, sort of short to medium duration increases in how much allergen someone can tolerate. Years ago, the FDA accepted as a sort of clinical trial readout the change in where someone reacts before treatment in terms of the amount of allergen to after treatment, and hopefully that amount is higher, and that's considered in most studies what's called the primary endpoint. And so that made really the clinical pathway feasible for work to happen in the field, but it does have a lot of limitations. And one of the things we don't really have, really good randomized controlled trial data on is long-term, truly sustained benefit or something that we might think of as remission. So in several studies, as the field advanced, there were at least looks at, what happens if you stop therapy for a few months. And we call that, sustained unresponsiveness for people who are still, able to tolerate a higher amount of allergen after a few months of avoidance. But it's a little unclear even now, how that corresponds exactly to what we do see from observational data and retrospective data sets, that some people after going through OIT really do, become functionally non-allergic, similar to the changes that we see in people that maybe naturally outgrow the allergy. But what remains unclear is exactly how big an effect OIT has on that, because we know some of those people will outgrow their allergy on their own anyway. But, so feasibility is sort of always creeps into how, a clinical treatment is developed. And sometimes that affects our ability to generalize that, more broadly. But what we have learned, and my lab, does work along these lines and many others as well, about immunotherapy, is some of the changes that we see in the immune response during immunotherapy. And I'll highlight a couple of things that we think, may be functionally relevant. It's often difficult to rigorously prove mechanism in human studies, but this, I would say, is where some of the most consistent, reproducible observations are, and they're backed up in many cases with corresponding data from models or in vitro to suggest functional relevance. And the two things I'll highlight are really, this uh, reduction in the kind of allergy skewed T cell subset that's called Th2A in the literature, and the induction of non-IgE antibodies, IgG, especially IgG4, and over time, the decrease in IgE antibody. So IgE, of course, is the class of antibody that arms mast cells to then be poised to have, induce immediate responses in the context of secondary exposure. So, you've become allergic, you've made IgE, and now you have an accidental ingestion to your allergen, and you have, anaphylaxis or other immediate hypersensitivity presentations. And so, over the years, there have been a number of things that people have looked at as potential adjuncts or helpers to OIT, including things that are sort of well upstream, you might think of it, in this pathway leading to allergic disease, or things that are further downstream. We're going to get to omalizumab. That's super downstream, all the way here, blocking IgE. But some of the more upstream things have been interesting. And I wanted to highlight in particular some recent data that comes from the combination of dupilumab with OIT. And so, because this was just recently published, we were part of this study here at Mass General. This was looking at, this was an industry-sponsored study by Regeneron looking at the combination of dupilumab with oral immunotherapy. Patients were randomized to get dupilumab versus placebo for 4 weeks before starting OIT, and for an additional, 4 to 6 months, in some cases longer, depending on how quickly people could build up to maintenance along with OIT. And then they had another food challenge. So food challenge at the beginning to define what that low level of reactivity is before patients are treated. Food challenge again after to see how much it's increased, if any. And then we'll come to I'm going to really just focus for time on this primary endpoint. And the upshot is that, although the impact was relatively modest, there was a statistically significant increase in the number of people, the percentage of people that could tolerate that initial primary endpoint challenge if they had been assigned to the dupilumab group versus the placebo plus OIT group. So just to highlight, none of the patients at baseline could tolerate this much. So OIT alone, doing what it does, increases the percentage of people that could pass a 2-gram challenge. But when combined with dupilumab, that percentage was modestly increased. So, that's, I think, maybe the best example in a short time that we have together, Carlos, of one of the ways in which, the people are trying to um, adjunct OIT currently, and in addition to omalizumab.
That's great. No, that's very helpful and put it in the context of the pathway of how these interventions could play a role in the upstream pathway where OIT could actually see a benefit. It's excellent. You
Right.
talked a little bit about dupilumab in combination with OIT, especially just in the initial phase of the response. Um, Switching to where I think we get a lot more questions because it's now the first biologic approved for food allergy treatments, omalizumab or Xolair.
Yeah.
Uh, can you tell us a bit more? I know you participated in great trials with Xolair and the Outmatch study where you can see Xolair alone or also in combination with OIT, and there's different phases of that study. Can you educate our audience and myself about the different phases of the study and how is Xolair could be working also and the benefit in combination with OIT?
Sure. So just to come back to this pathway for a second and remind us all, right, that Xolair's target is what we would describe as being very distal or downstream in this allergic pathway, right? So Xolair is targeting IgE, binding to IgE, inhibiting that IgE's ability to be, taken up or bound by these allergy cells called mast cells. These are the bad actors that, upon secondary allergen exposure, release histamine and other mediators that cause the signs and symptoms of anaphylaxis. So, what the design of Outmatch looked like is here. Um, and it's a complicated study. Believe it or not, this is the schema that was presented to participants who were considering consenting and participating in the trial. So it was a lot to talk people through. Um, I'll remind you that stage one, which I think probably many people in the audience have had some exposure to, is really the stage that produced the data that led to the FDA's decision to approve Xolair as a treatment for food allergy, as a treatment in the sense of um, protection from anaphylaxis due to accidental ingestions. So omalizumab as monotherapy for prevention of reactions due to accidents. That is what is FDA approved today. Any other use beyond that is off label, as it's called. And this was the arm of the study. Patients randomized to either active drug or placebo baseline, after a baseline challenge showing that they were reactive to multiple foods, and then repeating that food challenge after, a course of treatment with omalizumab, either every 2 or 4 weeks using a fairly standard dosing table, very similar to the way Xolair was dosed in asthma, extending it a little bit into patients with somewhat higher IgE, but still limiting the study enrollment a little bit because some of our patients have IgEs that fell outside of the range that was in the dosing table for Xolair. Stage two then was sort of an interesting and now published. You can find that. I should have put the reference here. I apologize, but recently published comparison of outcomes for patients essentially getting OIT after a short course of omalizumab versus omalizumab alone. So some data on the combination, but really looking at Xolair's ability to facilitate OIT. And I'm not going to focus as much on this actually because I'm going to show some other data, but essentially the upshot of that was that when measured against the primary endpoint and as what's called an intent to treat analysis, in other words, everyone that starts counts, people did better with omalizumab alone than they did with omalizumab facilitated active OIT, actually. And that was largely, I think, fair to say, because a number of people, are unable to tolerate OIT. So even though Xolair supported them being able to start it pretty quickly, ultimately a fairly significant percentage of people fell out. In stage three, which I want to talk about a little bit more actually, because I think this is sort of new and exciting and something that, I'm talking with, patients about already, as we, look forward to getting more data, is asking the question, how many patients who have demonstrated ability to tolerate, at least a minimum significant amount of their allergen are able to then consume that allergen in a somewhat normal kind of everyday way, even as Xolair or omalizumab is then withdrawn. Um, and so I thought it'd be interesting to this was presented at national meetings, and there'll be, more coming out on this soon. So I thought I might whet the appetite of people. First of all, just to remind you of that primary endpoint from stage one, these were the data that the FDA acted on. So just to sort of set the benchmark, what it showed essentially was that for most foods, about 2/3 of people were able to consume a cumulative total of 1,000 milligrams of their allergen if they were in the active omalizumab group. And you can see the much lower numbers of those in the placebo group, and this was highly significant and led to the FDA's action. And it was also looked at in terms of, how many people could consume at least two of the three qualifying allergens that they came into the study avoiding. How many people could tolerate three, not regularly. This is just that primary endpoint. How many people could tolerate on a food challenge at least 1 gram of foods. That's this first group here and the first sort of part of the survival curve. And then as the amount of allergen goes up, you see more and more people drop off. But you can see, almost half of the individuals in the study could tolerate really a serving size of at least two of the foods that they were allergic to on entry. And more like a quarter could tolerate all three of the foods that qualified them for being randomized into the study. Okay. Feel free to interrupt me, Carlos, on behalf of the audience if there's something that kind of came through unclear. All right.
No, this is great. This is great.
So in stage three, as I mentioned, this is where people were, if they tolerated at least a minimum amount on their post-treatment food challenge, they were given the opportunity to begin consuming allergen regularly.
Wayne, you got muted. We cannot hear you right now.
How's that?
That's better.
So what's been published, and the reference is down here below at the 2025 national meeting, is the 60 individuals that were in the stage one open label extension group. And, there were 82% of them that met the post-treatment bar for being able to be tolerating at least enough allergen that they could begin daily consumption with at least one food. And then success in this stage of the study was defined as those individuals that were continuing to consume a median daily amount of 300 milligrams after a year. And just to remind everyone here for a second, the omalizumab, was discontinued, essentially at the same time that they achieved their ability to tolerate at least that minimum amount. So Xolair's half-life is a few weeks, and so they begin consuming, these daily amounts as Xolair is beginning to wane away from their system. And so overall about 50% of individuals, a little bit more in the cases of milk and egg and wheat, although the numbers for wheat are really small, were continuing to consume those foods after a year. And I think, I wanted to highlight this because I think it's on the one hand, I think it's sort of a maybe glass half full, glass half empty, but something to potentially build on. On the one hand, you could say, well, gee, half of individuals, that's not much. On the other hand, maybe pretty impressive given that these individuals did not go through, months of OIT like buildup. They literally tolerated an amount in front of us that we thought was enough for it to be safe for them to begin consuming daily, and then began doing that, even as from that day forward, Xolair was being cleared from their system. And so in some ways, I think it's actually, fairly impressive. So, there's a lot still to come out of the Outmatch trial. It was really a privilege to be a part of it. This was, sort of an amazing consortium and collaboration led by Bob Wood at Johns Hopkins, and with support both from the NIH and from industry. And there's going to be more and more dividends still to come from that study for a little while. I want to pivot though, to some of the limitations and kind of highlight an older study about Xolair and its ability to facilitate allergen introduction. This was a smaller Canadian study, a few hundred people. It was actually an open label study, so take that with a grain of salt in terms of the strength of the data. But I think it's really interesting and really creative by this group of investigators to try to address some real world questions about omalizumab. I mentioned to you that quite a few people are actually excluded from omalizumab dosing by the dosing table that we currently use. Because Xolair is, targeting free IgE, and because its best understood mechanism is to reduce IgE's ability to bind to those allergy cells, those mast cells, it's dosed using a combination of IgE concentration in the body and total body weight. So it's essentially dosed trying to target enough drug to make sure that it's almost all of the person's IgE is bound by drug and unavailable to then have access to the mast cells. And that's not easy. That's a high bar. And also, it then takes some time for IgE to come off those mast cells. And that's part of why, one of the clinical limitations that we face with Xolair is we have to advise people need to be on it for, ideally 16 weeks, prior to expected efficacy. It may, have a quicker onset than that, but we know for sure it takes time. That the strength of the interaction, the affinity of the interaction of IgE to its receptor on mast cells is very high, and and we're limited by the time point that we have clinical trial data to support. But this was a study, that dosed people off that table substantially, and got a very sort of a wider range with less relationship between the amount of drug and their total body IgE. And what was interesting here, think of these as sort of like survival curves. We're looking at the percentage of people as they're going through a series of increasing doses of allergen all in one day. We're looking at the percent that have success getting to each of the doses. Okay, so the way to read this is as you go from left to right, we start off at 100% of people tolerating nothing, right? And then as you go across, fewer and fewer people tolerated the 1,200 milligrams on day one. And what the authors have done is to organize that survival percentage by stratifying it against the level of Xolair normalized to their own IgE. So in other words, um, because they didn't really stick to the dosing table, they have a range of people getting different amounts of drug to total IgE. And what you see is there's really no relationship when stratifying the patients in this way. There's no clear pattern of who is more likely to get to 1200 versus who is not. But in the lower half in panel B, same individuals simply stratified by their drug to body weight dose. And here, very interestingly, we see a striking stratification in the outcome as well. So those individuals who received a higher amount of drug per kilogram of body weight did significantly better than those that did not. Um, and so this raises a few questions about, other mechanisms by which Xolair may be acting. And what I'm excited about is that there are now 3 centers, ours being one of them, that are currently reevaluating in new clinical trials aspects of Xolair dosing and whether we need to limit it to those individuals who have IgE below a certain upper limit, and as well whether we should be dosing it just by body weight instead of normalizing to total IgE. So my point here really being a lot to learn still, about a drug like Xolair.
That's fascinating. Great data. People are very excited. I know we talked to a lot of patients and parents who are very excited about the possibilities with Xolair. So especially that of if it's normalized to body weight versus IgE, seems very promising. Um, you talk about a little bit about dupilumab, OIT, Xolair with OIT. What else are you seeing out there from your perspective that you think may have an impact or positive or not as exciting?
I think it's a really exciting time in the field, Carlos, because there are a lot of emerging therapies. We're seeing vaccine-like studies looking at virus-like particles, peptide vaccine. That there's a study that's going to read out soon. Um, Orally available mast cell inhibitors, a lot of things. And I think what is interesting in this time right now is, we have kind of in some ways a couple of domains. Um, those treatments that you might think of as sort of being agnostic to what you're allergic to, omalizumab being, the currently available um, drug in that category, but there are other anti-IgEs coming up on their heels. There are, small molecule inhibitors of enzymes that are important in the function of mast cells. So drugs that can suppress mast cells like remibrutinib. There's a phase 3 clinical trial set to start for food allergy for that drug, a drug that's, been just recently approved for chronic urticaria, and I think is really promising. And then you have, these innovations in the allergen specific space. I think you had Dr. Kim a couple months ago talking about sublingual immunotherapy, a lot of excitement around that. I mentioned already virus-like particles, a couple companies pursuing interventions with agents like that basically display allergens in a manner that is more rational and likely to induce, or at least hypothesize to induce, a non-allergic and potentially beneficial immune response, peptide vaccine. And actually, I'm embarrassed that I left an important category off here. I now realize as an example, and that's sort of the passive immunity. So, taking advantage of what we think one of the important mechanisms of OIT is, which is to induce protective non-IgE antibodies and give individuals those antibodies that have been engineered in a form of what's called passive immunity. So not stimulating the person's immune system to produce those themselves, but actually supplying it. So we have these kind of 2 domains of allergen specific on the right, allergen agnostic on the left, but what I think is even potentially more exciting to come as we have more and more of these things, hopefully, that emerge successfully in clinical trial and in their development, um, the opportunity to look at more ways in which immunotherapy type interventions may be appropriately combined with um, uh, treatments that specifically target aspects of the immune response. So just as we're seeing now with the combination of OIT and Xolair, as we've seen with the combination of OIT and dupilumab, I think that the field will open up for more opportunities to look at these kinds of combinations as we have more and more things available and proven to be safe and effective. So, I think what we might be missing beyond that, Carlos, is, we've spent a lot of time, if you come back here to this pathway, on things kind of generally speaking, mostly on this end. Um, but really there has been some work combining microbiome interventions with OIT, some promising, some less so, but a lot of, I think, fundamental understanding that we look forward to, in part through the efforts of FOSSI on the role, for example, of sensory neurons and their interactions with the immune system, that I look forward to the day that that's going to open up to more therapeutic options as well. So those are where I kind of left the slides, Carlos, but look forward to talking more and addressing questions from the audience as well.
Excellent, excellent. Thank you, Wayne. Really appreciate it. And a lot of good information, which I hope it helps our audience answer some of the questions that I always hear. Before we jump into the Q&A, briefly, I want to remind people, we always get the questions of how can I help donate, host an event, follow us in social media. We're a nonprofit organization, and our mission it's really to advance the research by working with smart people like Dr. Wayne Schreffler. And so your support in all these aspects allow us to push the boundaries in food allergy research. So, and I want to remind people that our next webinar series or next webinar, it's going to be with Dr. Michael Prisner, and that will be March 17th. So please register and join us. We have a few questions, Wayne. I have a lot of questions, but I'll just I'll ask just one. You talk about upstream, downstream. Dupilumab being more upstream than Xolair. Xolair is really interacting with the IgEs. Dupilumab more with the IL-4 and that pathway. What are your thoughts about why you're seeing the responses from Xolair and dupilumab being different? Or do you have any thoughts? Are there any possibilities? Do you see any possibilities for dupilumab to be used in the future more regularly for food allergies?
Yeah, I mean, I I do, and the clinical trial data I shared, I think were received to some extent with a little bit of disappointment that, there was a few things we didn't see, right? We didn't see a suppression of immediate hypersensitivity reactions in those patients. So even though there was a modestly higher success rate, I think it was a short time on dupilumab before allergen was introduced. I think I and others in the field wonder if a little more time. Dupilumab being a little bit more upstream, certainly can target a number of sort of the immune circuits, let's call them, that we think are really relevant, that including suppressing the effects of those Th2A cells, which is one of the most reproducible kind of observations we see in terms of immune changes corresponding to better OIT outcomes in adjuvanted or adjuncted OIT settings. And so and we also see, we know for sure, even going all the way back to the early asthma trials, that IgE levels certainly fall for patients, on dupilumab, and that there's a really, I think, interesting thread in the literature showing how important the kind of constant turnover and replenishment of IgE memory is dependent on new IL-4 signaling because it seems to reside in IgG class switched B cells. And so while there certainly are long-lived IgE secreting plasma cells, over time on dupilumab, Ig levels fall in a way that's not easy to explain from any impact directly on memory cells. So, um, on the other hand, clinically, there's been observations repeatedly that even in individuals who have, declines in IgE on monotherapy with dupilumab, in many cases they remain, clinically allergic on challenge. I will share with you anecdotally, I've certainly seen exceptions of that, people that appear to have outgrown their allergy that I wouldn't have expected. But of course, the field is full of people smarter than me thinking that because of their anecdotal observations, they have insight. So we always have to be, you know, skeptical about that kind of data. But I think to me that study is deserving of further refinement and replication. Perhaps longer treatment time on dupilumab before allergen introduction. And I think critically, the combination of dupilumab with antigen exposure, whether it's OIT, sublingual immunotherapy, or other routes, my hypothesis would be that might have benefit. I think that's still a good hypothesis to pursue further. Um, and the thing about Xolair, Carlos, is in many ways, or other anti-IgE targeting pathways, and this could be true of targeting mast cells as well. I think the bar still remains pretty high for showing that IgE blockade leads to a fundamentally different immune response to allergen exposure. So it definitely, makes like clinical OIT better tolerated. And I think, there are, patients in clinics, um, sort of building on the observation from stage 3 outmatch and trying to introduce allergens in a sort of less burdensome way than traditional OIT. So I think omalizumab, is already proving, use in that way. Again, off-label, consult with your doctor, all the caveats. But whether it fundamentally is improving those outcomes, really modulating, the way that individual's immune system is responding to allergen, I think is, remains to be shown and is, for my money, less likely than something that's further upstream in these pathways.
Yeah, and there's just so many unknowns still. I'm fascinated by the observation of what I would have thought would be more specific of normalizing to the IgE levels rather than body weight makes it's almost the opposite way, right? Indicating that there may be other mechanisms that we haven't explored since. I know we're short in time, so I just want to address at least one of the questions that we have here from our audience, from Hannah. If you had a kid who failed a single food challenge that triggered anaphylaxis and elevated multiple cross-reactive IgEs over the next few years, would you wait for science to catch up or take action sooner? And what kind of action? I think you probably get these kinds of questions from your patients. So, what are your thoughts?
I do, and they're questions that I really think have to be discussed one-on-one with your provider, and that's not intended as kind of the cop out, that it probably sounds like, but it really is so true. I mean, I think, and this is an exciting thing that there are more options, even though it can be frustrating, kind of trying to get definitive advice on which way to go. And there is that gap, of generalizing clinical trial data to patient experience to begin with, let alone a question like this that's really starting to go beyond even the clinical trial data that's available, right? So it's a shared decision. I think, uh, things like omalizumab do have the benefit of being around a long time and with a lot of safety data associated with them. And I think that helps, when you're trying to um, come to a decision together with a family and you're thinking about the risks and benefits. That's certainly something that's in the positive category. I lean, toward being more interventionist where we can and where I think it's safe to do so. But I also really try to not get too far ahead of our skis in terms of where we have data to really guide us. So
Excellent, excellent, Wayne. Well, I know we've come to an end in respect for respecting the time for everyone. Wayne, it's great to always talk to you and learn from you. Uh, One of the beauties, I think, of what FAST investigators are doing is actually approaching the problem from different angles, whether it being direct cellular and molecular mechanisms in the labs, but also in the clinic. And the data from the clinic, like the example you gave today, Wayne, really not only provides alternative and therapeutic alternatives to patients, but it also helps people understand better what happens in humans when they're treated with interventional clinical studies, which are so important because it drives a lot of information and it's a communication back and forth between what happens in the lab and in the clinic. So thanks so much for this, Wayne. A lot to learn and a lot to explore and go deep into mechanisms so that we can generate better therapies in the future. Thanks to
our
everyone for
pleasure.
joining
All
today
right.
and a pleasure, Wayne. Thanks again.
Thanks.